Hello, ma'am.
Candida, you're audible.
Yes, ma'am. Thank you, ma'am. Also, am I visible?
No.
Not visible, but audible. Maybe. Yeah, correct.
Okay, thank you, ma'am. Thank you so much.
Now, ma'am? Now? Should I speak? Is it okay now, ma'am? You're able to hear? Oh.
We can hear
Yes,
you.
yes, Richard. Jyoti? Jyoti,
Yes, ma'am.
ask her if we can start.
Yeah. Good morning, Dr. Tatu.
Samson, just check whether Dr. Tatu is muted.
Yes, ma'am. Sir has muted himself. He was able to speak when he joined in. Sir, I'll just switch on your microphone from my end, if that's okay. Sir, can you say something?
Dr. Tatu, can you hear us?
There is it. Just 1 second. Can you give me 2 minutes?
Yes.
2 minutes? Yeah.
Yeah.
I'll just mute myself.
Yeah, you can begin.
Okay. Good morning to everyone present here. In light of the global pandemic of the coronavirus, there has never been a better time to explore the fields of biological sciences in hopes of seeking advancement to the technologies and information already present at our disposal. I, Calderda Pearl de Souza, cordially welcome you all to the lecture workshop by the Science Academies on OMIC Sciences in the Times of COVID Pandemic, organized by the Department of Microbiology, St. Joseph's College, Autonomous, Bengaluru. Prayer is a surge of the heart. It is a simple look turned toward heaven. It is a cry of recognition and of love, embracing both trial and joy, said Saint Therese of Lisieux. I request Jennifer and group to invoke God's blessings with the prayer song.
Call me out upon the waters, the great unknown, the fleet may fail. And there I find you in the mystery, in ocean's deep, my faith will stand. So I will call upon your name, and keep my eyes above the waves. When oceans rise, my soul will rest in your embrace. For I am yours, and you are mine. Grace abounds in deepest waters. Your sovereign hand will be my guide. Where feet may fail and fear surrounds me, you never fail, and you won't start now. So I will call upon your name, and keep my eyes above the waves. When oceans rise, my soul will rest in your embrace. For I am yours, and you are mine.
Thank you for your mellifluous singing. A house with its doors always open is a home. There is little in life as reassuring as a genuine welcome. I request Dr. Beatrice Sequeira, HOD of Microbiology, to welcome the gathering.
Good morning and a very warm welcome to one and all present here. It's a privilege for me, on behalf of the staff and students of the Department of Microbiology, St. Joseph's College, Autonomous, Bengaluru, to welcome each and every one of you for the second sponsored Science Academy's two-day lecture workshop titled OMIC Science in the Times of COVID Pandemic. This workshop has been planned and designed by all of us for you, dear participants, and you will be exposed to a variety of topics and speakers who will be sharing their experiences. We cordially welcome our convener, Dr. Utpal Tatu, President of Proteomics Society of India and Professor from the Department of Biochemistry, Indian Institute of Science, Bengaluru. We thank you for all your support and deeply appreciate your presence here today. We are delighted to have among us our Rector, Father Dr. Swiba De Silva, SJ. A very warm welcome, Father. One of the chief motivators of all intellectual activities that takes place on the campus is our beloved principal, Father Dr. Victor Lobo, S.J. We wholeheartedly welcome you and invite you to be a part of our deliberations today. It's a pleasure to welcome the Registrar, Dr. Melvin Colasso, and the Vice Principals of different blocks of our college. A hearty welcome to all of you. My warmest greetings to all those gathered here for this intellectual lecture workshop. My colleagues from the department, my dear students, and participants from other colleges. A warm and hearty welcome to one and all. I hope this platform provided will enlighten, inspire all of you. Please do make the best of the opportunities provided, interact with the speakers, and take with you the main objective of learning and progressing to uphold the cause of science. We are indeed grateful and thank the Indian Academy of Sciences for sponsoring this workshop. Wishing this workshop a success, I now take this opportunity to thank all the participants and once again extend a warm welcome. Have a fruitful and enriching experience. Thank you.
Thank you, ma'am, for your warm words of welcome. Our principal, Dr. Father Victor Lobo S.J., is a man of precision and meticulous efforts who stops at nothing short of excellence. He is the pillar of strength and support that the college leans on. I request you, Father, to address the gathering.
Yes, sir. It's all done.
Dear Father Rector, Registrar, Vice Principals, members of the staff, the Department of Microbiology, guests, and my dear students, a very good morning and a hearty welcome to you to the Science Academy's lecture workshop titled OMIC Sciences in the Times of COVID Pandemic, organized by the Department of Microbiology, St. Joseph's College Autonomous, Bangalore. A hearty welcome to Dr. Utpal Tatu, the convener of the workshop, the members of the faculty, and dear students. It is said, welcome the challenges, look for the opportunities in every situation to learn and grow in wisdom. We as educators are entrusted with the responsibility of keeping the flame of learning ablaze. These trying times have compelled us to embrace new avenues. We have welcomed digital learning, and that has opened a plethora of options where learning is not restricted by physical barriers. Knowledge knows no bounds, and such workshops serve as the perfect medium to enlighten multiple minds at once. Microorganisms, minuscule entities with enormous impact. We stand witness to the power of these tiny forms and the havoc they can wreak. Although this crisis is owing to the coronavirus, not all microbes are always harmful. At such pressing times, it is thus imperative that we see both sides of the coin. This workshop, with its impressive lineup of speakers, intends to take us all on a journey with these diverse life forms and attempts to comprehend them better. We would like to place on record our immense gratitude to the Indian Academy of Sciences Bangalore, for providing such an excellent platform for learning and currently allowing the conductance of workshops on virtual platforms to benefit innumerable young minds. On behalf of the management of St. Joseph's College, I wish good luck to the Department of Microbiology and all the participants. May these 2 days be an enriching and memorable experience for everyone. Take care and stay safe. Thank you very much.
Thank you, Father, for your words of insight and acuity. I now request Vice Principal of the Science Block, Assistant Professor, Department of Microbiology, and the coordinator of the event, Dr. Jyoti, to present an overview of the lecture workshop.
Good morning, all of you. St. Joseph's College, a 130-year-old institution, 38-year-old institution, is one of the biggest Jesuit educational institutions in India. The college was conferred with the status of College of Excellence by UGC in the year 2014 and was re-accredited with A++ grade and 3.74 by 4 CGPA in the fourth cycle of NAC. Academic excellence, social concern, and character formation are the three main objectives of St. Joseph's College. The Department of Microbiology started in the year 1988 and PG section in 1998. The department was awarded the DST FIST grant in 2017. With the able guidance of head of the department, dedicated staff members, and enthusiastic students, the department has achieved many laurels over the years. With the spirit of Majis, the department has organized various conferences and workshops all these years, and even during this lockdown, the management has motivated us to do more. I quote Sallust: "Harmony makes small things grow, lack of it makes great things decay." Every day we live and every meal we eat, we influence the great microbial organ inside us, for better or for worse. The gut microbiome, along with secreted proteins and metabolites, has emerged as an important factor affecting human health and disease. In the last decade, clinicians have been able to modify their patients' microbial ecologies by replacing a bad microbiome with a good one through a fecal transplant. These findings have further facilitated microbiome targeted drug delivery and efforts to improve human health management. The recent development of omics approaches, including microbiome profiling, shotgun metagenomics, metatranscriptomics, metaproteomics, metabolomics, and microbiomics, has enabled efficient characterization of microbial communities. However, little is known about the regulation of microbes in the gut ecosystem and the underlying interactions with the host. Scientists are trying to understand what microbes are there, what they encode, what they produce as communication signals, and how these change over time. Keeping all these advancements and challenges in the field of microbiome research, this 2 days lecture workshop was carefully crafted by our workshop convener, Dr. Utpal Tatu, Professor, Department of Biochemistry, Indian Institute of Science. His lab is one of the first labs in Karnataka where ICMR approved reverse transcriptase based COVID test kits were developed. In these 2 days, you will be taken through various aspects of omic sciences, genomics of pathogens, antibiotic resistance, social behavior of bacteria, microbiome of eye, gut microbiome and liver functions, and so on. So, make use of these 2 days of Indian Academy of Sciences sponsored lecture workshop to interact with scientists and surgeons to enlighten your knowledge and understanding. All the best and have a great day.
Thank you, ma'am, for the insightful overview. We are all indeed eager to experience today's events. With that, I hand over the platform to the moderator, Madiha, student of second year MSc Microbiology.
Thank you, Kaljida. I welcome all the participants again to day 1, session 1. I hope everyone's geared up for an extremely engaging lineup of lectures today from some of the very best in the field. Prophet Muhammad once said, there is a done to an animal. Our first speaker of the day, Dr. Utpal Tatu, is not just an accomplished scientist, but also social entrepreneur. Dr. Utpal Tatu is a celebrated professor at the Indian Institute of Science and obtained his PhD from the same. He pursued his postdoctoral research at Yale School of Medicine, New Haven, Connecticut, and went on to become an assistant professor there. His most notable research dwells into the neglected infectious diseases such as amoebiasis, babiosis, candidiasis, giardiasis, malaria, rabies, toxoplasmosis, and trypanosomiasis. With all this, he aims to develop diagnostics, medicines, and therapeutic drugs to make them widely affordable. With over 2 decades of experience in global health, he continues to implement his research expertise to improve human and animal health with social entrepreneurial initiative Equine Biotech Labs. This initiative, powered by the Indian Institute of Science and headed by him as a director, aims to improve and develop diagnostic and therapeutic approaches to zoonotic infections. He has received numerous accolades: Birla Science Prize, Bioscience Career Development Award from the DBT, and Ranbaxy Research Award, to name a few. He is an elected fellow of the Indian Academy of Sciences. He was formerly the president of both the Proteomics Society of India and vice president of the Indian Society of Mass Spectrometry. He is also on the editorial board member of the Parasitology journal published by the Cambridge Press. Given his experience, Professor Tatu's reputation is not limited to being nationwide, and he is on the research advisory boards of biotech companies in India and abroad. We welcome you, sir, and I request Dr. Utpal Tatu to deliver his talk on the subject that is the current need of the hour, developing a sensitive and accurate diagnostic for COVID-19. The floor is now yours, sir. I kindly request you to please share your slides. Meanwhile, I also request the participants, let's make this session an interactive and engaging one. If you have any questions, please feel free to post them in the comment section, and Dr. Utpal Tatu will be taking them at the end of his session. Thank you. Over to you, sir.
Okay. Thank you, Ms. Madina, for your kind introduction. Am I audible to everybody?
Yes, sir. You're audible. Yes, sir.
Okay. Thanks again, Ms. Madiha, for your very generous and kind introduction. I'd like to also thank Dr. Jyoti, who's a professor at St. Joseph's College, Department of Microbiology, for all her efforts in putting together this wonderful theme workshop sponsored by Academy of Sciences. I also thank Dr. Beatus, who's the HOD, Department of Microbiology, as well as all the honorable administrators and the leaders at St. Joseph College. I have not had opportunity to meet all of you in person, but have heard so much about you, and I'm eager to meet you one of these days. I've had long association with your college, and it's been a pleasure for me to mentor many of your postgraduates who have worked in the past with me and who have now gone on to pursue higher studies in other international levels, including at United States in very famous universities. It's indeed been a pleasure for me to interact with students in your college who have always appeared eager to learn, sincere, and wonderful cultural etiquettes. I look forward to my further interactions with students and faculty at your college. Thank you again for this opportunity. I'm very happy that faculty at Department of Microbiology have taken this initiative to bring to the student community a wonderful mix of lectures which touch upon contemporary aspects of diseases that surround us, and add to that some wonderful new advances that have taken place in the area of microbiology and in general in disease biology, to help students understand where the science is moving and where are the future opportunities. I thank Indian Academy of Sciences for giving me this opportunity to be the convener for this exciting event today. With these few words of introduction, I'll start my presentation today. I'm going to talk to you on the broad topic of global health and share with you some of my experiences in how my research laboratory, me as a person, and a social entrepreneurship initiative out of Indian Institute of Science have placed ourselves to try and make a small difference in the world where we are surrounded by challenges to our health in the form of diseases which existed in the past, which continue to challenge us, and newly emerging diseases. I will talk to you very briefly. Just 1 second. I think I'm getting a message. Audio has to be set right. I'm going to just try and see what I can do.
Dr. Tatu, your audio is fine. The issue has been rectified. You can continue with your presentation.
Very good. Thank you so much.
Yeah. Sir, if you don't mind, the pop-up is also visible for us. So could you just click the first one, the dash sign, so that will minimize the next one left back. Yeah. Left, sir, left. Yeah. So yes, sir. So this will help us see your slide better, more of it.
Excellent, excellent. All right, thank you very much. What you see in front of you is, of course, the title slide, but also the campus of Indian Institute of Science. And I always choose this purposefully to entice students from your renowned college to visit us, to interact with the faculty here, and to explore opportunities to work and participate in research programs here. I have been very successful in attracting many of the bright minds from your college, and I hope that more students will take advantage of moving here on a short time basis or on a long time basis. What I'm going to do today, and as Dr. Jyoti had already introduced, we are going to talk about omics science. I'm going to start my presentation with this, an article, a scan of an article which appeared in Time magazine several years ago now. But really, the article captured a very interesting facet of how science is now touching society. Magazine such as Time, which is not a science magazine, it's your general social journalism magazine. It carried a full-fledged article titled as Blood Work. They couldn't use the term genomics, proteomics, but they used the term blood work. And the article, as I'll very quickly show you, indicated that in today's world, and as if I may just quote the title of the article as it appears on your screen, blood work, finding new cures means cracking the body's complex proteins. So scientists are turning to big data. And that's really what the theme of today's workshop is going to be, the big data science. How are big data science, which includes genomics, proteomics, metagenomics, are helping address some of the most complex aspects of diseases that plague mankind. What this article highlighted was, with this image of drop of blood in the center and various human ailments which are indicated on the circumference of this drop, that it's possible with just a drop of blood to diagnose most complex diseases, which oftentimes include diseases of various organs of our body, but also cancers, and of course, including infectious diseases. One that's mentioned here is prion disease. But of course, it's just a representative, any infectious disease. If I may just turn to the next slide. The article quickly jumped onto the term proteomics, as is shown in the top panel of this article, second page of this article. It really used the term proteomics, which we think is a hardcore scientific term. But here it was making visible presence in Time magazine already 7 years ago. They called this Proteomics 101. What are the basics of proteomics? And what it said was that mapping the human genome helps scientists determine risk factors for diseases, and searching for proteins tells us about the state of the disease in patients now. And that's really what they said, that human genome and human genome sequence helps us predict the risks. But it's the proteins which really tell us the current state of the disease, whether the individual has the disease or not just about predicting whether the disease is going to happen. And that's really the main sort of step forward that this article helps make common people do, that yes, we have arrived in the field of genomics, but now it's time to herald into the age of proteomics. The figure at the top really shows what's very basic to science students, but important information for non-scientists and for people who are non-specialists, that nucleic acids, the genome, codes for various large amount of information through its genes, and also non-coding sequences, which can tell us predisposition to diseases based on the single nucleotide polymorphisms and mutations. But when these genes are expressed into proteins through transcription and translation, the proteins which are made are the workhorses. They are the ones which tell us the current state, and any anomaly in higher level expression, lower level expressions, absence of a protein or presence of a disease causing protein is indicative of the presence of a particular infection or a particular disease in human body. And this is what most scientists are trying to capture today, that can we look at the protein signatures at the level of the proteome and try and predict the current state of infections. In this last, I'm not going to show you the whole article, but what it also narrated is that this is going to impact the diagnostic industry. Diagnostic industry has existed for a long time, as we all know, from the time when the first blood test came out, very simple measurements of metabolites, measurement of ions, and so on. I mean, we know that this has been around for ages. But now it's taking a new turn because scientists are going to look deeper into what is present in a drop of blood through the most modern scientific methods of omics, which includes big data science, which I mentioned has aspects of genomics, and then of course proteomics, and now we are moving also towards new terms such as metabolomics. And then today's workshop, today and tomorrow, you will also hear about a new facet of this omics called metagenomics. Hopefully in future, I'll talk to you not just about metagenomics, but also about metaproteomics, which is yet another new field that is quickly making its presence felt in scientific circles. The summary of this article really was that this diagnostic industry is going to grow tremendously. And there is, of course, pharma industry and big players in medtech industry keeping a close watch on how to make this whole area efficient. I start always with something that appeals to common people, and that's why I chose this article to connect you with what the topic that we are going to talk about today, before we jump into the deep science of it. I will make stepwise inroads into the main theme of the conference today. And so this slide brings to you some basics or something that will appeal to you in terms of what basic science and genomics and proteomics has to offer. What this slide shows to you is we are in the post-genomic era. Biology in the post-genomic era has changed the way people, changed the way scientists are practicing science, changed the way scientists are using new tools, and changed the way how these new tools are going to reach to common man, going to reach where it's needed the most. What the slide highlights is the cover pages of various scientific magazines that all of you are very much aware of, the ones which we aspire to publish our research findings into, such as Nature and Science, some of the best magazines. And the cover pages of several of these highlighted the genome sequencing efforts of various organisms, be it plants, animals, or microorganisms. What's shown here, and as my cursor moves, you'll see that genome of corn, maize, was sequenced many, many years ago. Genomes of Plasmodium falciparum, which is a microorganism that causes malarial parasite, was sequenced many, many years ago. Another infection called trypanosoma was also sequenced and made it on the cover page of Science magazine. And then again, higher organisms such as, be it honeybee, be it Drosophila, or even mammals such as monkeys, the macaques. And then again, people have sequenced the whole genome of larger mammals such as horses. And I don't need to emphasize this, but all of you are aware the human genome itself is now published and very extensively studied and becomes a part and parcel of all research activities that we carry out in research institutions. What is available to us today is at the nucleotide level information about the gene sequences present in these which are displayed to you on the slide, but many more organisms that have been sequenced. The genome sequencing technology has progressed so rapidly and has become so convenient that it is possible for us to sequence genomes very efficiently in short period of time and with relatively less financial inputs. As a result, this field has created information that we are unable to actually comprehend yet. In the past, the science was actually dominated by what we did not know. In the past, science meant what is it that we did not know, we do not know. But today, in the post-genomic era, science has the problem of how do we interpret the data? We have this data, but how do we make sense out of it? And that's really the big challenge in the post-genomic era. Because of the presence of large amount of genome sequence information, we are surrounded by interesting information that we are trying to gradually learn to decode, and we are trying to make sense out of. And this is what the challenge of big data science is. And we continue to make progress, and that's really what I think proteomics is all about, that we are now moving from genome to predict the proteomes and study the proteomes and try and make important insights into biomarker development and diagnostics of diseases. And hopefully this will also lead into better therapeutics. I want to just end this conversation, a brief conversation that of the introductory aspects of omics, with a real life example. What you see in front of you is image of the patient of the future. This is already 10 years ago that a gentleman by name Larry Smarr, American gentleman at the West Coast in California, who happens to be in the IT field. He's a data expert. He knows how to use hardware and software to analyze data, but not in biology. But he got so impressed with what he was reading about big data science, including the article that I showed you in the previous slides from magazines such as Time, that he said, hey, I want to apply my computer skills and my computational prediction methods to decode this data. So he started talking to experts in this field and started examining genome sequences, proteomes, and metabolomes. To an extent that he started experimenting with his own biological samples, be it blood sample, urine sample, sweat sample, and whatnot. He started sequencing his own genome. He started interpreting single nucleotide polymorphisms. He started looking at blood work, as was earlier described in Time magazine, and also trying to see that can he predict his own body physiology. Larry Small was considered a crazy guy. But hey, I think West Coast is known to be, very liberal, open to new ideas, and people are, embrace, what you might call disruptive ideas, the big breakthroughs. They're by no means traditional in the way they look at new aspects of culture, new aspects of science, and new aspects of society. And here was one example where people actually, on the one hand, challenged Larry Small that, hey, are you wasting your time with all of this? One fine day, Larry Small walks into his physician's office, his doctor, and says that, Doc, I've been reading my metabolome and my proteome for last several weeks, and I see these perturbations in my profile. I will not get into the specifics of the data, but he told Doc that, look, I think I'm going to have some gastrointestinal trouble. There seems to be some problem with my bowels. And the doc examined him very carefully because he was not only a doctor, but also a family doctor, and as a result, an old friend. So he humored his client and his friend by saying that I'll examine you completely and thoroughly. And at the end of those 15 minutes of physical examination and pretty, I think, intense analysis, he said that, Larry, just go back home. It's interesting what you're doing, but don't push it. I don't think it's making sense what you're telling me. I don't know how to read this data, but what the bottom line that you're saying doesn't make sense. And Larry went back home, but sure enough, in a few weeks, he developed gastrointestinal trouble. And this is an interesting story. It sounds dramatic, but it's true. And that's why I've shown the picture of the gentleman, and it's become, it went viral in the United States. And he did come down with what he predicted he's going to have. And he went back to the doc and said that, look, this has happened, and his predictions were correct. So really, the point is the patient of the future is going to have more information than the medical professionals who treat them because of the access to data, because of the access to information that can be found out from small amount of samples. And today it's possible that any of us could get our whole genome sequenced through facilities and services that are available all over the world. We can get metabolome generated, we can look at proteome generated. This is all possible, and this is how the science is moving, that common people will have access to data, and then they will, many of them are going to try and make sense out of it. I'm going to jump into now a little bit more specifics, but before that, I just take a pause to see if there were any comments or questions or queries about what I've talked. If anybody wants to post a query, you're most welcome to do it. I also want to very quickly ask if it's possible for you to let me know how much more time I have in this session.
Sir, you can go on till 10:30, sir.
Excellent. Okay. Sorry for interruption, but I think it's better for me to keep track of time.
No problem.
So I think by now, most of you are prepared to jump into the science of what we are going to talk about with the information that I've given you. And I hope this will not, this prelude will help you feel more excited about what we are going to discuss. This appears to be a busy slide, but I'm not going to bother you with the right panel and the bottom panel. You just have to pay attention on the left top. What I'm showing you here is what you already know. And as a science student, you already know DNA codes for RNA. RNA gives rise to proteins. And then there are the small molecules which are circulating in our body, which carry out many biochemical functions. Even when I was a student, we learned that DNA it undergoes replication to copy itself, and central dogma, of course, that it gives rise to RNA by transcription. RNAs get translated into proteins, and those are the workhorses of the body, and they oftentimes generate metabolites through biochemical reactions such as Krebs cycles and glycolysis and whatnot in the body. And all of you science students are familiar with that. And we were happy to learn that and understand that. But as a science student today, and as teachers who are teaching science today, you have to be familiar not just with this basic self-centered dogma, but also what's shown on the right side of this picture. The terms of genomics, transcriptomics, proteomics, and metabolomics. If we were to have a face-to-face interaction, I would ask the students, tell me what do you understand by the term genome? The term genes and genetics and genetic engineering have been around for a while, but all of a sudden, out of nowhere, the term genome found its way into textbooks, into scientific discussions, into research, and into research presentation. Quietly enough, this has made important impact, not knowing where it originated. So it's interesting to scratch our heads and say that we are running a conference on genomics and proteomics, but where did these terms come from? Of course, I won't have too much of time to dwell into it, but it's a very interesting anecdote, and we must think about this. Now we are also excited about this term, genomics. We'll leave that discussion on the side. I'll leave that as a curiosity, sort of something for students to pursue and think about. But I will move on to say that today's student has to understand not just the fact that DNA can code for RNA, but understand what is there in the DNA. Of course, genes are there in the DNA, but that's not enough. We need to know how many genes are there in the DNA. In the previous slide, I showed you the genome sequences of large numbers of organisms. So that makes it possible for us now to say that a particular organism, such as in Homo sapiens, there are about 25,000 genes. So everything that we do, everything that our body is capable of doing and functioning can be explained by an interplay of 25,000 genes. Of course, that's oversimplifying things, because things that part of the DNA, part of the genetic material that does not code for genes is also equally important, and that's the significantly bigger part of the genome, which does not necessarily code for genes and as a result proteins, but they are the non-coding sequences that often govern the regulatory aspects in our body. And we are only beginning to learn the complexities of these non-coding regions. But hey, we want to think about what we know. We know that there are 25 to 30,000 genes. What are these genes capable of doing? They are capable of producing. When transcribed, they can produce as many as 100,000 mRNAs, 100,000 transcripts that are capable of undergoing translation to give rise to 1 million kinds of proteins. And that's the level of complexity that's present in the genome, and particularly human genome, that about 20 to 30,000 genes, and we don't know the precise number yet. We are getting better day by day in defining the number of genes because of very good bioinformatics that's available today. But we know that if all these genes, even 25,000 genes were to be expressed, we would have approximately 100,000 transcripts. And these 100,000 transcripts would give rise to 1 million proteins. You might wonder why, how come 25,000 genes give rise to multiple number of transcripts? How does more than one transcript arise? And that's because of all the molecular jugglery that is encoded in our genome. There are lots of changes that happen in terms of regulations. As a result, you can have one gene giving rise to multiple transcripts, alternative splicing. You have editing processes that happen, and that's some of the things that give rise to multiplication of numbers, 100,000 transcripts, and then it results in even more number of proteins, because while you can code an mRNA and translate it into a protein, the protein can be post-translationally modified in various different ways. So if you include all the post-translational modifications of a particular protein, and each modified protein as a separate entity, and indeed which it is, because with just one modification event such as phosphorylation, you can change Jekyll into Hyde. A protein which is beautifully functioning and carrying out its function by a mere single phosphorylation can become completely inactive and vice versa. As a result, each modification can change the protein, and that's why scientists treat each modification as a separate protein entity, and that's how you end up with a million different kinds of proteins. And now that's really what the challenge for this generation of students and scientists is, that we need to define numbers. It's not good enough to say that there are proteins and there is this protein and there is that protein, but we have to see that how do we deal with a million proteins in the cell? Well, the good news for us is that not all the million proteins are produced at the same time in complex organisms like human. These are theoretical numbers, because at any point of time, maybe a small portion of these may be produced. But 20,000, depending on which tissue, which part of the body, which part of the cell you're talking about. As a result, there are subproteomes. There's a liver proteome, there is kidney proteome, there is cardiac proteome, there is neuroproteome. All of these different organs don't produce all the proteins at the same time. There is tissue-specific expression. And so, but even then the numbers are large and we are aware of those numbers. Finally, I want to end this slide by talking about this last arrow, which goes from proteins to metabolites. That's a whole new field of metabolomics that's come around now. And look now, as the numbers from genes to transcripts to proteins continue to increase exponentially or significantly, but the metabolomes are relatively small. Of course, this number 2,400 is a highly underestimated number, because this slide was made at a time when metabolomics was just starting. But now the number has been revised, and it appears that since we are talking about human body having about 25 to 30,000 genes, the metabolome might comprise of about 10 to 15,000 metabolites, or maybe at most 20,000 metabolites. Those are the number of metabolites that regulate processes in our body. And that's really what is interesting. As you know, metabolites don't arise, they cannot be coded by genes. These are small molecules such as glucose, acetyl-CoA, be it pyruvic acid, be it malic acid, be it various of these metabolites that we are aware of. They arise and they get converted into each other through the actions of certain enzymes. Those are the ones that are coded by genes. But oftentimes they can be. So what I'm trying to say is that while you can estimate the number of RNAs and number of proteins from the gene sequences and the genome sequence, you cannot predict the metabolome from genome. That has to be experimentally determined. And today you have tools such as mass spectrometry and nuclear magnetic resonance, which help you to study metabolites and metabolomics. That's whole level of complexity, which is difficult for even scientists to fathom. And that's going to be an exciting new phase of omics sciences in the years to come. Somewhere amidst this is the term metagenome, which is somewhat different. And I think I will not going to touch upon it today, but we'll talk about as we progress. And some of the experts that I think I have volunteered to give talks, which include some of the best surgeons in the country. They also are researchers. They will talk to you a bit about metagenome subsequently. At this point now, I want to change tracks. I've given you a lot of information about the main topic, omics. Now you're prepared with the tools. But where do we apply these tools? Looking at what's going on around us today, I think we must apply these tools to remain alive. We don't know if we go to, today, I think the way we are dealing with COVID pandemic, I think there is a whole new appreciation for human life. So many things that we took for granted in the past, we do not take for granted. We are so much more careful about the way we carry out our day-to-day business. As you all know, the level of hygiene, the level of care, the level of interpersonal interactions, all of this has gone to a whole different level. We are so careful because we are not sure. None of us are sure what will be tomorrow like. And why has this happened? And this slide tries to bring to you something that we have always ignored. How do disease outbreaks happen? And I want to remind all of you that disease outbreaks have happened because we have neglected important part of human interaction with its environment. And this is what I'm going to talk about in the next few minutes. And that will sum up and would become a great platform for all the topics that are going to be talked today. I want to remind all of you that all the diseases that have challenged mankind in the last 10 years, and if I were in a face-to-face meeting, I would have asked students to tell me what diseases have they heard of, and the answers would have been Ebola, the answer would have been avian flu, the answer would have been Zika, the answer would have been Nipah virus, and of course, all of us are still in the midst of the grip of COVID. What's common among all of these infections? What's common among these infections is that all of these infections have come from animals or birds. And that's what I want to emphasize in this part of the talk. And then somewhere we'll bridge the first part of omics with the challenges that we are facing today. That how can we use what we have learned through omics to apply to diseases that are emerging and diseases that we are facing today. The disease outbreaks, and this is where I'm showing you on this picture, map of India and animals. All the infections that we talked about a few minutes back have emerged from one animal or a bird, some animal or the bird. Those diseases that are listed are not written on the slide, but those are the ones that we are most aware of, and we have suffered them in the past few months, past few years, and we are currently suffering from the challenge of COVID-19, which has appeared from bats, as we understand today. Again, a mammal, bat is a mammal, but it is a different, completely different part of the animal kingdom. So what has happened? What has happened in the last 10 to 20 years is that the way we as human beings, the way we have interacted with the environment needs to be corrected. Of course, many of us eat meat from animal sources, be it birds or all of this. We interact with these animals also because we love animals. If we are farmers, we are in the animal industry, we drink milk, all different ways we interact with animals to consuming their products and also interacting with them as animal lovers. We like to keep pets. Many of us have dogs and cats at home. Some of us have agricultural farms, and we have cattle in our backyard. We are also, as human beings, interacting aggressively with our environment. We want to live in, we want to have farmhouses in the wilderness. So we will cut the trees, buy acres of land, and rich people like to create farmhouses. And that's good because we want to be close to nature. But in the process, we are wiping out the ecosystem there, and we are coming closer to the life that pre-existed there. And in other words, we have intruded in a new place, where human activities did not happen in the past. And that's the social aspect that needs to be brought. That many of the infections that we are seeing today have actually jumped, and most of them have jumped. And you'll be surprised to hear that as many as 70% of infections suffered by human beings are transmitted from animals in one form or the other. And many of those are listed on the slides. These are they are continually present around us, be it anthrax, trypanosomosis, theileriosis, babesiosis. They typically are there in animals, but they can jump, and many of them have jumped species barrier and have come into humans. And so also brucella, which is very common zoonotic infection. And then there are various other viral infections. And needless to emphasize, COVID has come from bats. So the point that I'm coming to is that today, let's not wait for another infection to jump from animals to humans and then try to deal with it. Let us address the root of the problem, the foundation of the problem, which is keeping animals healthy. Making sure that animals are healthy, they do not have infections, and then hopefully then many of these infections will not transmit into humans. And that's why the term that was described earlier today was neglected infectious diseases, emerging infectious diseases. Neglected term means that these diseases have been around, but for some reason, pharma industries have not paid enough attention to it, for the reason that there is no economic or financial benefit for an industry to work on developing treatments for such infections, because these infections lie in parts of the world where people cannot afford expensive medicine, in part of the population that cannot afford to pay large amount of money for treatment of such infections such as malaria, tuberculosis, and many of the other infections that I talked to you about. And COVID is it's an example in life. It's around us and it's a problem. The vast population in countries like India cannot even afford to get themselves tested. The test itself costs a couple of thousand rupees. And they're probably earning 1,000 rupees per month. How are they going to spend that just for a test? And that is the kind of problem that we have to deal with. And needless to emphasize that, there are other infections such as rabies, which continue to challenge us and have been around. Lots of humans, as we speak today, are also dying of rabies, which is transmitted from infected dogs. Now, where do dogs get it? I will leave that for you to think. We know that dogs transmit rabies to humans, but where do dogs get it? Again, a question that I want you to think about. I will not answer this question, but that's another area of research that I'm extremely interested in and engaged in. I hope I have triggered some thought process in your mind about contemporary issues, the problems that surround us, and how science can help us do that. I'm going to skip some of these slides because I've made my point that interactions with animals and wildlife and the domesticated animals, and mixing of all of this creates an ecosystem that helps viruses and bacteria and parasites thrive and undergo changes and coming out to challenge humans in a new form altogether, with various mutations that make them more aggressive in transmitting, various mutations that enhance their virulence. And then human beings are at loss because we were not ready for this strain of the virus, this form of antibiotic resistant bug, which subsequently Professor Dipankar Chatterji, a senior professor from Indian Institute of Science, is going to discuss with you, I think, in the talk that follows this introductory talk. With that, I want to move to the last important aspects of antimicrobial resistance, because that again is the way humans have interacted with the environment and used some of the traditional antibiotics, which of course have helped us cure diarrhea, cholera, and many other infections and non-infectious diseases. But then widespread use of these antibiotics has resulted in antimicrobial resistance, because the bugs have continually got exposed to this drug more than we should have exposed them, and the bugs have mutated and found ways to resist them. And this is what has now further complicated the system. With this, now I'm ready to jump into the main topic, but I've created enough background. What I decided to do was, instead of showing you a lot of technical data, I've equipped you with the basics of omics science. So even if you were not a science student, you will now have hopefully a better appreciation for what is going on in scientific research today and what problems we are facing, and how current COVID infection, as well as many infections that we have experienced in the last few years, have emerged from animals, and how we need to address the root of the problem, the foundation of the problem. Only then we can have a healthy future. As a research group at Indian Institute of Science, this is what I did about 15 years ago. I've been at Indian Institute of Science now for 24 years, but 15 years ago, I decided that it's not enough for a researcher to just publish papers. And I developed a whole map of how I will try and pursue my science, at least make an effort at a small level as an academician, and try to make an impact. And with that, I will end my talk with this slide, that I have for the past several years, I'm focusing on neglected infectious diseases, emerging infectious diseases, which include some of what we talked about, but also include, parasites such as malaria, trypanosomosis, trichomonosis, giardiasis, amoebiasis, and so on. More recently, I have dabbled into rabies and COVID. How can a researcher not work on COVID? Because we have to do whatever small effort that we can. The idea in the lab then is, on the one hand, we train PhD students and project assistants and trainees and interns who come from wonderful organizations and institutions like yours. We use fundamental aspects, multidisciplinary aspects of sciences. And of course, the main job as researchers is to publish papers, because we have to highlight our research findings at the international level. But in addition, of course, what I have done is to try and enhance or extrapolate my science in the field by accessing clinical samples from infected patients and animals, apply the big data sciences, and I'm going to talk to you in my subsequent talks, next generation sequencing and genomics and proteomics, and then try and make sense out of this to develop biomarkers and develop diagnostic tools and drugs. This is really the pipeline, and this was what I had envisaged. But what I'll show you is that part of the efforts have paid off. For a scientist, it's always hard to go through the whole pipeline of converting a research finding into a product. But I'm trying, I'm making an effort, and it's not too late. And many of my colleagues at Institute of Science and all over the country and all over the world are trying to do the same. We're trying to make science into some useful products in terms of diagnostics and drugs. And I have had a small success, a very small, tiny success. I do not want to exaggerate it, but it's a quest that one has to continually work on. And I'm going to show you how we have today test for COVID through the efforts that we have made. And that's, as Dr. Jyoti and Dr. Beatas briefly touched upon, that's a government approved test that's now available through Indian Institute of Science. And I'm going to narrate that story in the next, in my next talk. I was given time until 10:30 to launch, to kick off this conference, and I hope I've done some small justice. I apologize if I have disappointed you by not showing you a lot of scientific data. But I will stop at this point. It's indeed been a pleasure for me to be part of this conference, and I'll be available. I'll be attending many talks today, but I'm available even subsequently for students to reach out, for teachers and professors to reach out to give opportunities to students, and also I enjoy interacting with young, bright minds to create opportunities for all of that. Thank you so very much for your kind patience.
Thank you so much, sir, for that gripping session. You've definitely done justice to the first talk of the day. We have a few questions which we'll be taking now. If anyone has questions even now after this, you can please post it in the comment section. So the first question is by Dr. Syed Wajid. Can the metabolites... Yeah, can you hear me, sir? Hello?
Hello?
Can you hear me, sir? Hello?
Yes. I was expected to stop at 10:30, so I hope I'm not too late or not too early.
No, sir, it's perfect. You can hear me, right, sir?
Yes, I can.
Okay, so thank you for that session, sir. You've definitely done a lot of justice to the first session of the day. So we'll be taking some questions if that's all right with you, sir.
Absolutely, absolutely. It'll be a pleasure, of course.
Okay. The first question is by Dr. Syed Wajid. What molecular techniques are currently used to trace out the genes responsible for metabolite synthesis? Only if we know the genes responsible, we may explore the options of metabolic engineering.
That's right. So just repeat the first part of the question.
What molecular techniques are currently used to trace out the genes responsible for metabolite synthesis?
Okay. All right. Very good. Very good question. I think. Thank you, Dr. Wajid, for this intriguing and thoughtful question. The question tells me that you are deeply into science and you have been thinking about challenges such as this in the past. What Dr. Wajid is talking about is genetic disorders. There are so many genetic disorders that human beings suffer from because of mutations or various other genetic changes that happen in the body because of exposure to various things, including mutagens and so on, and because of genetic defects. I will not get into that, but I think if there was an example in front of us, it would have been better, but I can think of many examples of metabolic diseases, and Marfan syndrome is one. There is various inborn errors of metabolism, including Gaucher's disease and phenylketonuria, would be an ideal example of what Dr. Wajid is talking about, and which most students have studied in the basic textbooks. These are diseases because a particular enzyme in a particular metabolic pathway that is essential for important conversion of one metabolite into other. For example, in case of phenylketonuria. And that's really what Dr. Wajid is talking about, that when this gene is mutated, it results in accumulation of a precursor which does not get converted into a product. And what do you do in such cases? What are the solutions to such genetic defects? And the answer to that question is that conventionally, about a few years ago, we did not have corrections for such genetic defects. People have been toying with the idea of gene therapy. And there's a lot of research that's happened in this, and people are thinking that you could replace a normal healthy copy of the gene with the defective gene in the body, and lots of experiments have taken place. It has met with some success. Gene therapy is a solution, a potential solution to this problem, and it has met with some success, and there are examples where this has helped, but it has not been as successful, as robustly successful as would have liked. But modern new methods that we are hearing about now, people are talking about CRISPR-Cas. As you know, just last week, Nobel Prize was announced in the field of chemistry for two wonderful women scientists who laid the foundation to CRISPR-Cas based gene manipulation. And these are the methods. Unfortunately, I won't have time to get into it. These are the methods which hold tremendous promise in converting a defective gene into either removing the defective gene or adding a corrected copy of the one, and in general, in treatment of such diseases which result in malfunction of an enzyme because of a mutation and then manifesting itself in the form of a disease. So yeah, the answer would be that yes, CRISPR-Cas and gene therapy are the ways to correct such metabolic diseases. And today, the only way these diseases are being treated are through management. We are not able to cure the disease, but we have to learn to manage these diseases through diet management, through lifestyle management, and so on. But time has come when we can now hope that gene therapy and gene manipulation through CRISPR-Cas will make it possible to make irreversible changes at the genetic level to cure this. I hope I've addressed this question, Dr. Wajid, if you want to follow up.
Thank you, sir. The second question is also by Dr. Syed Wajid. Can the metabolites be also biotransformed substances that are present in the environment, or the nutrient substances that utilize from the environment?
Can the metabolites, may I request you to repeat the question?
Sure, sir. Can the metabolites be also biotransformed substances that are present in the environment, or the nutrient substances they utilize from the environment?
Okay, all right. So the first question, can the metabolites be transformed into useful substances? Yes, absolutely. I think it's something more relevant for a plant world, I think, where many metabolites can be converted into useful substances. And that's why we have some traditional drugs be it a drug for malaria, the quinines and chloroquine, and then many drugs for cancer such as vinblastine and taxols. These are all plant products, and many of them are plant secondary metabolites which have undergone transformation to give us, either in the lab and to give us the active form of the drug. That's part of the answer, that yes, some of the metabolites, and these are the secondary metabolite examples that have been there with us for generations, that have become drugs and are currently the best drugs that are available for some of these infections. But these methods have now moving more aggressively. People are thinking about metabolic engineering, where they can actually enhance production of some of the useful metabolites. That is one new scientific development that people are trying to do, metabolic engineering. And then, of course, some of these drugs get converted into other products in the environment. That part of the question I didn't quite understand. I apologize but I hope I have addressed at least part of the question.
Yes, sir. Thank you. So the next question is by me, Madiha. I was just curious if from these pandemics that have started from non-domestic animals like bats, is it possible that we can predict pandemics such as this or prevent it in the future? Because these animals are not domesticated. Is there a way we can control diseases that they are infected by?
Absolutely. I mean, you have touched the most important part of my presentation. And my emphasis was really that let's not wait for outbreaks to happen. Let's catch the problem in time and at the source. And we have to worry about infections of animals. Even if they don't affect us today, they may tomorrow. And which is what Madiha nicely captured in her question. Really, I think if you think about it, on the one hand, we think we appear to care for this animal world, but we have not done enough. And the glaring example is, the whole, I'm sorry, I'm going into a non-scientific aspect of this answer, is that, we have the whole veterinary world, a veterinary college, a veterinary doctor, veterinary treatments. And then on the other side, we have the human doctor, the human health, and all of that. The very fact that there are these two different arms of medical practices, veterinary medical practice and human, which are disconnected, is really where the problem is. I'm sure all of you understand this, that today qualified medical personnel who look into animal health are totally different from those who look into human health. And in fact, there is a social hierarchy in this, and whole different domains of life. They never talk to each other. And that's really what scientists are trying to change. And I'm making my own small efforts at whatever levels possible at a One Health program that has now captured attention all over the world, emphasizes that these are not two separate issues. We have to address them on the same platform. And this is where research laboratories like me are doing that. We look at human infections and animal infections together because there is a common dynamics, often described as One Health. And hopefully such research programs will help bridge. We also organize conferences where veterinary doctors and medical doctors, human medical doctors, talk to each other and engage. We also engage in research programs where internationally and nationally we have collaborations with veterinary doctors on the one hand and human medical doctors on the other. And COVID is one example where we are doing some small efforts in this way. But Madiha, you have touched the main question, and I tried to address this by highlighting the fact that animal health has been ignored, neglected, has been undermined, and we need to change that.
Yes, sir. Thank you. The next question, I think, is a follow-up by Nishi Mehta. Does this mean it is possible to somehow control the spillover of infections by managing human interactions with the environment? If so, what might be the ways we can achieve them?
Very, very good question. And this was also part of Madiha's question, that is it possible to predict? It's possible to do something about it. I mean, we can't completely stop. We can't claim that we will not have infections in the future, but we can do something to control it. We can do something to minimize these chances. And the way to do that, I mean, it's obvious to all of us now that we're talking about it, that we need to routinely monitor animal health. Just as we monitor human health, right? Even if we are perfectly healthy, we undergo routine medical checks. Isn't that true? We go to a doctor and do the blood work. It is prescribed so many different tests, amounting so much expenses, but we readily do it to make sure that, oh, I'm not an early stage diabetic. I hope I'm not, my heart is functioning properly. I hope I don't have, kidney problem, and so on and so forth. Right? All the time, people are going to medical laboratories and doctors to ensure that their health is okay. But on the other hand, for animals, until the problem stares at us, when the animal is obviously ill, we do not bat an eyelid, unless it's a very dear pet. And this is what has to change. We need to have routine diagnostics, what you might call preventive diagnostics. And that's, so now I'm boiling the problem, I'm sort of distilling the problem into Rishi's question. And yeah, I will address that. Yes, routine testing of animals, be it wild or domestic, has to happen at much more aggressive level. We could have prevented influenza outbreaks possibly if we had done it. We could have prevented, many of the infections that we are seeing today if we had done it. Corona infection was around us. It was lurking around us for decades. I probably should emphasize that as researchers, we were working on coronavirus infection diagnosis in animals even before people had heard of COVID-19. And that's what is interesting. And then again, this infection now became more aggressive because of mutations and made its presence in the form of COVID-19. And that's when we suddenly woke up to this reality that we wish if we had addressed coronavirus and infections in animals more aggressively, but that didn't. So one way to do that is what I already said, that address animal infections more aggressively. There needs to be platforms at government level, at social level, all of that to make that happen. And we are doing that. We are trying to promote that idea, and we are actually practically doing it in our research programs and through the social entrepreneurship that we have done. We reach out to farmers, to animal lovers, to animal industries, and asking them to monitor animal health on a routine manner at the same level of accuracy, same level of rigor that we do in human health. That's one. Now, prediction, yes. I think that's where we are still not there. If there is an animal infection, can we predict that it's going to actually become a human infection in future? No, I think the answer is currently we don't know that. But by doing the measures that I talked about, we can try and minimize the chances. We will never be able to stop interacting with animals. We cannot stop interacting with animals completely, but we need to interact with them safely by making sure that the animals that we are interacting with are healthy and have been tested. And let's start that at an individual level. Each of us, if we have pets at home, let's start with our own pet. That's really how we need to start. It's just preaching and giving lectures like I'm giving is not going to help. We need to start action. And that's going to happen only when we do what we can do at individual level by some simple solutions that are provided, that I've thought about. And we are doing it. And I'm happy to talk about it when there's more time. Thank you for that question.
Thank you, sir. So the next question is by Arun Kumar E. Will it be a breakthrough if we analyze the metabolics of gut microbiome? If yes, will biopharma industries shift their focus on manufacturing the gut metabolic compounds?
Very good question. And thank you, Arun, for that question, because you're now taking us into the topic of gut microbiome and microbiota, which is going to be discussed partly today by some really expert surgeons from the city, but also by medical practitioners. And then I will conclude it tomorrow in the concluding session. Thank you for bringing that topic up in your question. I will only be able to answer part of the question, which is, yes, is gut microbiota going to help us understand metabolomes and so on? Yes, that's already happening. And microbiota, many of you, of course, know gut microbiota means the the microbes which exist in our biological system, be it gut or all parts of the body, oral microbiota, eye microbiota, gastrointestinal microbiota, all parts of our body have microbiota, which is useful. As they say, it's not a human body. Actually, human cells are being carried by microbial cells, because in our body, there are more microbial cells than our own cells. So these are all different different ways of articulating this issue. And yes, science has advanced significantly today that gut microbiota is being used to treat and to prevent problems in human body. I'll give you an example, and a beautiful example, and a fascinating example in something that I'm myself engaged in. There is very quickly, even problems such as kidney failure, and this doesn't come obviously to us, that even problems such as kidney failure can be addressed through microbiota, through manipulation of microbiota. And there are people are now claiming to have probiotic products that will help control kidney failure or management of kidney failure. To that extent, science has progressed, and there are these products already out there in the market, which are under the title of supplements, dietary supplements, wherein the capsules consisting of certain microbes which are in the dried form, which upon consumption of the capsule will enter the gut and stabilize there on continual consumption of these tablets. These are microbes which actually help process many of the toxins which are not processed by the kidney because of its malfunction, and thereby help get rid of toxins which accumulate in people who have kidney failure. So really, this is a wonderful example of what is actually out there today, even kidney ailments. And I know this very well at first hand, because I was involved as a scientific consultant in development of this product by a company which is now released this product, which is out there available to all of us, and many kidney failure patients consume it. But that's an example and part of the answer to Arun's question.
Thank you, sir. We'll take the last two questions now. The next question is by Vaibhav Kulkarni. In order to meet up with the demands of the growing population, farmers are forced to pump farm animals with antibiotics. Do you think that this could lead to a worse pandemic in the future?
Thank you, Vaibhav. Again, very thoughtful question, and again touches upon something that I passingly mentioned, antimicrobial resistance. And you are very right. I think this is a time bomb waiting to explode. International bodies are emphasizing this, and there is so much talk about antimicrobial resistance. And now suddenly our attention has shifted to COVID and the pandemic. But antimicrobial resistance is what is going to result in creation of more robust bugs, which are going to come and stare at our face. So yes, you're absolutely right. The animal industry, and especially including poultry industry, are using lots of antibiotics, and that's not the level at which we are trying to intervene. We are also trying to examine poultry feed, cattle feed, the food that is given to these animals and birds, to make sure that they are free of such antibiotics. Wherever somebody comes forward to us, we are able to provide such services to create a standard that, look, what you're feeding to animals should be devoid of such harmful things. Sometimes inadvertently, sometimes intentionally, these are given to make sure that these infections don't happen. The infections don't happen in animals, but overusing antibiotics is creating a whole set of new problems. And countries like India have become notorious for overuse of antibiotics. And the problem, the whole world looks at India and they are pointing fingers at India, saying that we are promoting antimicrobial resistance because in our country, there is no regulation on the use of antibiotics. We can walk into a pharmaceutical shop and buy a strip of erythromycin without a prescription. And that is what is creating this problem. Yes, so thank you for bringing that out, and certainly your point is very valid.
Thank you, sir. The next question is by Larissa Gomes. With regard to patient self-diagnosis, in the example mentioned about Larry, how do you differentiate if the gastric problem was actually caused due to his predisposition or to pre-occurring the disease, or it was his mental state of interpreting his own data that led him to believe that he would suffer so? A case of mind over matter?
Yeah, right. I think it's always interesting, the psychosomatic problems. We always there is a basis to that. But in this case, of course, this example I chose because it is an example, and this gentleman actually did come down with inflammatory bowel disease. There's no question about it. He was treated for it, and because it was caught in time, it was managed very well. So there's no question that but there is a case in point there. Psychosomatic disorders is a reality. Many times we imagine something, and then that itself causes a problem. That is a reality, and we have to be aware of it. But the fact is that the sciences that we are talking about give you values, and that is scientific quantitation and evidences which are very hard, which are very reliable, and then there's no element of psychological angle there. So yes, but having said that, I will re-emphasize that your point is valid, that many times human ailments arise out of psychological fear. And today, I think we are also having a bit of that in COVID. There is so much of fear of COVID-19 that oftentimes the moment we get a little bit of scratchy throat, we are running to a lab to get ourselves tested for COVID-19, while it may be a routine bacterial infection that we all suffer one time or the other. But today, if I have a sore throat or a scratch, I will actually be so nervous that I will run to a COVID lab, I'll get myself tested, and the doctor himself or herself will ask me to get admitted for something that we would have totally ignored and managed by gargling or inhaling steam a few years back. This is the point that you have tried to make, a very valid point, that there is a balance that we need to strike here, and fear factor is a big thing in medical field. Thank you for bringing that up.
Thank you for that, sir. We have a couple more questions, I think more, but due to the time constraint, we will wind up the first session. But lucky for us, sir is also with us for tomorrow's session. So if time permits, sir can take them tomorrow. Thank you again, sir, for your first session. It was really, really interesting. And for the questions as well, participants, your questions have been really helpful and given us all a better understanding of the topic. With this,
thank you.
Thank you so much, sir. With this, we have come to the end of our first session. I kindly request our participants to please stay tuned for our next session, which is an equally pertinent subject by Dr. Dipankar Chatterji. Our next speaker of the day, Dr. Dipankar Chatterji, is also a professor at Indian Institute of Science. Professor Chatterji specializes in the field of molecular biology and biophysical chemistry. Professor Chatterji received his PhD from Indian Institute of Science in molecular biology and then went on to become a research associate at SUNY at Stony Brook, New York. Professor Chatterji returned to India in 1983 and held the position of Scientist to Deputy Director at CCMB Hyderabad. Here, Professor Chatterji established a group of scientists who worked on the regulation of gene expression in prokaryotes. In fact, his group was one of the few who showed inter-domain interactions in the transcription factor sigma 70 controls the maturation of enzyme RNA polymerase, as well as the role of omega subunit in the enzyme. Professor Chatterji has received his doctorate from Jadavpur University in 2018. Professor Chatterji has received numerous awards, some of which include the Homi Bhabha Fellowship, Shanti Swarup Bhatnagar Prize for Biological Sciences, Millennium Gold Medal at National Science Congress Pune, Ranbaxy Research Award. He was also awarded Padma Shri, the fourth highest civilian award in the Republic of India by the President of India. Professor Chatterji has over 200 publications to his name. But his achievements don't just end here. Professor Chatterji has authored the book Basics of Molecular Recognition, published by CRC Press. To add to the feathers in his cap, Professor Chatterji has a US patent titled Mycobacterium segmatis grown under carbon depletion conditions as a model of Mycobacterium tuberculosis under latency towards drug screening. Professor Chatterji later shifted his attention to the survival strategies of E. coli under nutritional stress called stringent response, and showed through a series of publications that RNA polymerase is a target for the stringent factor in E. coli. There was a long-standing debate on this issue, which was solved with this work, and now the model is well accepted. Professor Chatterji would like to continue to work in two areas in the near future. First one will be the crosstalk between two secondary messengers like tetraguanosine phosphate and cyclin-dependent GMP during starvation. They would like to continue work with transcriptional regulation in mycobacterium under nutritional stress conditions and during stationary phase. Professor Chatterji has guided 31 PhD students and continues to be an educator and guide at the Indian Institute of Science. What an honor it is to have you amongst us here today, sir. I now request Professor Dipankar Chatterji to enlighten us about the very pressing subject, antibiotic resistance, a ticking time bomb. I request you to please share your screen, sir. I'd like to remind the participants again, if you have any questions, please feel free to post them in the comment section. Professor Chatterji will address them at the end of his session. Welcome, sir.
Am I audible?
Yes, sir, you are.
I can't see the share of the screen. What do I see? Sharing screen. Share desktop, yes.
Yes, sir, we can see your desktop now.
Okay, I'll now switch on my presentation. Okay. Can you see it?
Yes, sir, we can see it.
There's echo back coming. I don't know how to handle it. Is that okay now? Can I start?
Yes, sir, you can start. It's clear.
Okay, thank you very much for your kind introduction. Thank you to the organizer, particularly Jyoti, for putting me in this talk. I must tell you, I was a little hesitant from the beginning. She knows it very well because I don't work on COVID. It's a pretty new phenomena, but our work is very much involving in antibiotic resistance, bacterial survival under tough conditions, stringent conditions, and antibiotic resistance therefore becomes a very important area of research which is connected with COVID also. So what I'll do for next 1 hour or so to the audience, I must tell you that I'll try to tell you why I'm telling you this story today and what is the connection between the deadly disease which we are suffering today and the situation we are in now. Antibiotic resistance, a ticking time bomb, has been touched by Professor Utpal Tatu during the end of his talk. I heard a couple of slides, and some participant raised the question, very pertinent question on this. So therefore, I am not completely out of page or out of gear. So I'll start my presentation now. The connection between COVID and omics is the main focus of this presentation, of this workshop, which is sponsored by the Academy. It is not a new thing because when I was going through the Google, I found there is an app site for COVID COVID-related omics, and several publications are indeed coming in for this particular area. What are the connections with COVID and omics are very important. What people have done, they have done the total mass spectrometric, lipid profile, blood samples of the patients and their degree of severity. It will definitely help to shed light on viral infection and mechanism of action of drugs. However, one must remember, when we talk about a mechanism of action, we must talk about how we can handle a particular proliferation of a disease. And we have been very successful as a human race to handle many of the bacterial infection with the antibiotics, which I'll continue further in my presentation. But we are not so much successful with viral infection. As you all know, viral infection, they propagate on the basis of the host. So they try to manipulate the host, they try to mutate themselves, and therefore it is very difficult to handle them. But what is most important, there are several pathways which are very important to kill them. And those pathways require inhibitors, specific inhibitors to handle those infections. So as we proceed in my talks, you will see that the bacterial infection can be handled by antibiotics, the classical discovery about which I talk about it tomorrow in my second talk. But remember, all the research workers or the future research workers, all the students must remember one point. Any living species try to survive. The survival and propagation, multiplication is the basic hallmark of any living species. We want to survive, bacteria wants to survive, virus also wants to survive. So if you put pressure on them with antibiotics or environment or any other parameters, they'll try to change, they'll try to mutate, they'll try to generate resistance. So it is very difficult to handle them under pressure. So our vigil, our try to get an antibiotics or a method to kill them is a continuous search. There is no end to it. Today some antibiotics works, tomorrow we will not see any antibiotic working at all. Same one, because we develop resistance. And this fight is going on for ages, for thousands of years.
Before that, I'll just get a printout. Hello? So we can.
Excuse me.
I'll just take a printout. Once you have the final copy.
I think someone has to mute themselves. Please, everyone, make sure your mics are muted.
Can I continue?
Yes, sir, please.
I heard somebody is talking, that's why I stopped. Hello?
Please continue, sir. We can hear you.
Okay. So one has to really do some clever design how to handle the infections, both bacterial and viral, by playing around with the metabolic important pathways. But always remember, their main job is to survive. They are not come here to get killed. Nobody wants to get killed. So there are other possibilities, ways and means to survive. And I'll talk about it tomorrow also in my social behavior talk. And that is the greatest learning we learn from our experiment research. So what are the pathways one can struggle? And classical pathways come from this higher structural hierarchy. If you look at, there are several levels of organization of macromolecules of biological systems. We can start from monomeric unit of nucleotide, amino acids, sugars, carbohydrates, which make them second level of macromolecules, DNA, RNA, protein, cellulose, cellular membrane. Then we have a supramolecular assembly. Then we get the total organism. So anybody's guess where we can interfere. You can interfere with every step here. Every step we can interfere. But you have to look for which are the most important steps. Making a DNA, making a RNA, making a protein are very important steps. This is a central dogma of molecular biology. But many times, if you interfere with those with antibiotics or viral infection, what will happen? That this interaction will create a problem for them, but they will try to adjust to that. So therefore, you have to cleverly design a pathway. That pathway can be adjusted in a very classical way, like you interfere with it, but mutability, the chance of mutations are very rare. That doesn't happen all the time, and that's why the mutation starts. In the case of COVID infection, why this is relevant? This is a viral infection, and if you look at the newspaper, it is a knowledge now, everybody knows about it, it's very much talked about. You will see some drugs are being utilized, are being used, hydroxychloroquine, remdesivir, etc. And each pathway of these drugs are very well known. There is no inhibition for RNA polymerase, RNA-dependent RNA polymerase, the viral enzyme. And it was first thought of a very good target because of HIV infection, which is also RNA-dependent RNA polymerase. But the drug which is used for HIV, remdesivir, is partial success with OMICs, with COVID, including President Trump has been given remdesivir. But it is not a specific drug. So the classical approach for virus infection to control virus is and vaccine development, which is going on. There are so many candidate vaccines are coming, subunit vaccine through adenovirus and through RNA virus, RNA vaccines, and so many kind of things. So this is not the point of the talking here. And heat-killed virus is also there. Polio virus is a heat-killed virus. But I am trying to point out here what are the specific pathways we can look at. And I mentioned DNA and protein are the major important any pathways which we can target. But we ask the question, and I'll show you, I'll try to convince all the audience here, that there are other pathways which are equally important and perhaps not so mutable. All pathways are equally mutable or not, we do not know under pressure. So the central dogma pathways are mutable, they are prone to mutation, so they create resistance, but there are also which are not so much. So can we target those pathways? And I'll try to show them. This is a central dogma hypothesis in 1958. All the people who are attending this talk know about it. Beautiful papers, beautiful theory has come out of it. And there are possible conversion that are reported till now. But there are also several ways. People who are listening to this talk should read and should go to Google and try to search why the term dogma has been utilized for these pathways. What is so dogmatic about it? Is every path is reversible? It has been very well studied and very well published also. It's very recently. The reason behind that dogma has been used in the literature. But people thought that attaching inhibition to any of these steps, DNA to RNA, RNA to protein, whether one can generate very good inhibitor. And that inhibitor, therefore, all living species follow this. Bacteria do it their own pathways. Virus do it on the help of the host. It could be plant, it could be bacteria, it could be human being like COVID. So mammalian species. So therefore, if you impair with these pathways, you can kill the bacteria or the virus in this way. So central dogma, therefore, there are having controversies. There are RNA editing controversies, single gene may code for multiple proteins. There are mutations, there are silence mutation. We also work on those areas, but I won't talk about it now. So it goes on like this. So look at the inhibitors, and I'm coming slowly to a point where I talk about these inhibitors in a very great details, because we said that if you look at the omics studies, proteomics, lipidomics, look at the protein pattern, look at the lipid pattern, look at the other metabolites, small molecules which are coming up. Many of them have some various reasons to exist after infection, like COVID. And those existence can be impaired with their metabolic pathways if you know about it. So to know them, you have to know the basic unit from which they are made of. And if you know that, you can impair with that pathway. So therefore, you have deoxyribose, ribose, bases, nucleic acid bases. So each one of them. So you ask the question here that if we make a derivative of this pentose, derivative of this nucleic acid bases, derivative of the phosphate group attachment, can we make good inhibitor for the synthesis? Don't think of any different thing right now. What I said so far, that our focus is, our attention is on the ways to make an inhibitor which will counter the process of making all these molecules only by the bacteria and the virus upon infection, not by the host. So therefore we can distinguish between them and we can generate an inhibitor. See, if this is the arrangement of a phosphate group attached to the pentose sugar, sugar attached to the base. This is the basic unit of the nucleic acid structure, and this is the general structure. If we generate mutation or generate a base, some other position like this, then of course I can have an inhibitor. What I mean to say, that DNA makes RNA, RNA makes protein. And this DNA to RNA synthesis, the transcription process, how all the 4 bases nucleic acid are coming as a template directed pathway. These are textbook, and now first year BSc students will know this. If I now stop the formation of the nucleic acid polymerization on the template, how then can you stop the virus or bacterial infection? It's a very unique concept. How can I stop? I can stop by mimicking the nucleic acid bases so that they cannot propagate, they cannot synthesize further. Or I can stop by having a some inhibitor which will bind to the enzyme which is making this and not allow it to function further. So 2 ways. I can kill the enzyme, I can poison the catalysis. Enzyme is nothing but a catalysis. I can do a catalysis poisoning, or I can as a substrate which will go and replace the bases and act as inhibitor which will not propagate further. With this point, many inhibitors have been designed which will go into the DNA based sequences, which will go and bind to the major groove or minor groove of DNA, and it will alter it and then look at the translation helical parameter. I will not talk about it, but the classical drug, which is nothing to do with omics right now, it is about cancer, cisplatin, cis-dichlorodiamine platinum, which is a very potent inhibitor for cancerous cell. It is even today is being utilized in the hospital for some kind of cancers with a lot of toxicity. It's a very good chemotherapeutic agent. This cisplatin was discovered in 1974. Why? Because the platinum 2 gets into between 2 base pairs in a DNA chain. The distance between 2 consecutive base pairs in a bDNA is 3.4 helix, 3.4 angstrom. I have written that 3.4 angstrom or 3.4 nanometer, 34. This is a whole. So 2 bases is 3.4 angstrom. And cisplatin, the cis distance is 3.4 angstrom exactly. So they just slip in. If we slip in the base pair in between 2 base pairs, the replication of the DNA is inhibited. So these are the kind of approaches people take at the basic level, whether you can stop the replication. Can I have an inhibitor? I will show you some example, which will bind to the enzyme, inhibit it completely. So these are the part of the omic studies which are coming as a approach, drug approach for killing an enzyme, killing a virus or a bacterial multiplication. So you look at the stability of the base pairs, you look at the hydrogen bonding ability, you look at the alteration of the DNA structure, etc., and you go on continuously. So these are the different kind of base pair. I will not go very much into details. But at every of these Watson-Crick base pair or different kind of Hookstein base pair, which is for triple helix, this kind of base pair synthesis or the formation can be impaired with several inhibitors. And these are all good approaches for making a drug. So compaction of the DNA, therefore, is another very nice way to generate a bacterial infection to control it. Again, I'm talking about bacterial infection here mostly because that has been very well worked out. For viral infection, as I told you, so far mostly failures, except certain inhibitors I'll show you. Future slides I'll show you. Most of the people go for vaccine development, but that is a more logical approach than a chemical inhibitors, which is so successful for bacteria. So the condensation of the chromatin happens in the following way at different stage. Again, at each of these stage are responsible for the packing of the nucleic acid for making a chromatin, and therefore it becomes a very important area for us to study further. And you can control this packing by different small molecules inhibitors, and there also you can have a inhibition process working. This is a DNA different conformation. Why I show this? Because there are alterations here. I will not talk about the left-handed DNA because its natural existence is still questionable. But right-handed DNA, bDNA, has typical geometry as I showed you before. And little white fat DNA, aDNA, is also typical geometry. There are positions in the DNA molecules we have worked out, many of them. People know now their base pair tilt, their number of base pair per turn, and RNA double helix, double-stranded RNA adopt this structure. And their difference, there are 11 base pair per turn, there's 10 base pair per turn. So there are some differences here. And these differences are very finely utilized for generating inhibitors for the RNA synthesis and also ribosome maturation, which have been worked out extensively, because if you can impair with the ribosome maturation RNA inhibitors, you can interfere with the translation process from transcription process, RNA protein synthesis. So at every step, my whole last half an hour or so I'm speaking, my emphasis has been always to tell you what are the different ways the central dogma of molecular biology or the central part of the macromolecular synthesis can be impaired, can be handled with, without tinkering very much with the host system. But so far, the success in this case, in viral, is partial, is not as good as the antibiotics or other cases. Why is so?
Excuse
How
me,
do I
sir.
define an antibiotics? It is defined as a chemical that kills or inhibits the growth of a microorganism. And in the last speaker has spoken the answer to some questions on this basis, so I'll go ahead with the same thing. The target is an entity and process. Please, hello?
Yeah, sorry to interrupt. There is a small request. If you can move the icon that is in the center of your slide to the left, because it's covering up some of the slides.
This one?
The one in the center, sir. The one with the mic and the video icon.
1 minute.
On top, sir.
This one?
Yes, sir.
I'm sorry, I can't do that.
It's not. Okay, sir. If it's not happening, then no problem, sir.
Oh, this one you mean?
Yes, sir.
Sorry about it, but
No issues, sir. It was just a small request.
I read. Target and mode of action. A target is an entity and process which should have unique structural and functional property in the cell. For example, ribosome in protein synthesis, RNA polymerase in transcription, I mentioned about it. It is not the case always. Several targets are promiscuous, rendering the drug discovery slow. Means the target can be very different and we have a problem with that target. So it can be promiscuous. Sorry. Try to play around with it. Sorry, I lost the slide again.
No issues, sir.
No, I'm not being able to remove it.
We can see a slide now, sir.
Huh?
We can see a slide now.
No problem?
No problem, sir.
This is a very important point which is written here. Antibiotic resistance is a phenotype acquired by bacteria during prolonged treatment due to selection pressure, attributed by several factors like mutation in the antibiotic binding site or due to the other physiological changes. What do we mean by that? I told you at the beginning of the talk sometime back that antibiotic resistance is coming up because the bacteria wants to survive. So you put a bacteria, when you say, I'll tell you, in my laboratory or many laboratories all over the world, people do it. I want to generate a mutation against an antibiotic. I know rifampicin is antibiotic, it could be anything, against E. coli, Escherichia coli bacteria. So if I put, you see this experiment, when I first did it, I found it such a simple experiment, undergraduate student did it. But it has such profound implication that if I plate it, if I put bacterial colony in a agar plate, which is a source of survival of the bacteria, I will see different small colony of bacteria coming up. If I do it with antibiotic, say 100 microgram per ml or 50 microgram per ml concentration, I will see none. So what I do, I reduce the amount of bacteria. I go to 10 microgram per ml. I will see some colonies coming up. Take that colony, I replate, now I increase the concentration of antibiotic. I make it 50 microgram per ml. Next time I see few colonies. So I go by serial dilution like that. I can generate bacteria, finally a colony, I went up to 400 microgram per ml resistance of rifampicin, which I know exactly why it is happening, the mechanism. So if you go on treating the system with a heavy amount of bacterial antibiotic concentration, you will see some resistance colonies are coming up. And these resistance colonies are dangerous because they are surviving even under the presence of antibiotic. That is the whole problem of failure of antibiotic treatment. When rifampicin is a very strong antimicrobial RNA polymerase, so it inhibits transcription process. When it was discovered in 60s, people used to think that we are out of bacterial infection, and death due to tuberculosis came down. However, I am sorry, in this lecture I cannot draw a figure. Otherwise, I would have drawn a figure on the board. If I look at the survival, you will see that it gets killed, but the killing curve never touches x-axis. That means never becomes zero. Few of them will remain, and we call them persisters or latent infection, which survive under such bacterial infection concentration of antibiotics. Those are dangerous, and they developed over a period of time. So now if you travel in a crowded train in Mumbai or in a bus in Calcutta, you know the person around you, they are carrying such, 1 out of 4 carrying such latent bacteria. Their immunity is strong, they are surviving. But when old age, you become senior citizens, or you have other comorbidities, they'll come up in, they'll survive further. They'll resurface or resurrect. So this is the whole point. So the fight is all the time is happening. And this happened because of chemical mutation in the central dogma transcription process. So this is a very thought-provoking. I hope everybody can read it. Otherwise, I will try to read about it. Timeline of antibiotic resistance, antibiotic deployment. It's so pathetic. I took this from 2007. Now the situation is even worse. Look at this. Sulfonamides is developed in 1930. Antibiotic resistance observed in 1935. Penicillin in 1940 by 1945. Tetracycline in 1950 by 1955. Vancomycin, erythromycin, ampicillin, methicillin, cephalosporin, streptomycin. More and more stronger inhibitors we are getting, and more bottom line shows how the antibiotic deployment at the top. These are deployment of the antibiotics, and these are the resistance of ampicillin coming. So if ampicillin was sealed, is ampicillin employed in 1960. By 1975, resistance came. If 1995, when Linozyte 2000 was employed, by 2005, Linozyte has amtobicin now. So any medicine you develop, it becomes a disaster after 10 years because of bad use. We are buying antibiotics across the counter. Another thing is that antibiotics are expensive. So if you take 2 tablets, you get resistance and you think your fever has gone, you are okay. But that doesn't kill. To kill 100%, you need to use it. All the doctors will give you a dose depending upon your condition or weight, 3 into 5 days. There is a complete course. Tell everybody. When I give this lecture, I tell it to every person. Tell everybody in your neighborhood or your relatives to complete the course. If you don't complete the course, you become a resistant, a carrier. So 3 into 5 means you kill all the antibiotics, all the bacterial infection, and you are much more, what should I say, good citizen, that you are not infecting others. So that is the whole game. This fight is continuously going on. So in India, we are buying antibiotics without doctor's prescription. Less amount, we play around with that amount of concentration. As a result, we are killing the problem. TB is a classical example that we failed completely to control it with bacteria, with antibiotics, because we used as our wish as they go ahead. Now, this is a funny slide, but it's so tracking the antibiotic resistance. 2014 it came. Animal, chicken, is full of tablets, full of antibiotics. Any other meat products full of antibiotics. So even if you are a vegetarian, you have problem because there are other issues on that cases. But if you are a non-vegetarian, you're a meat eater or a fish eater, you are eating basically tons of antibiotics when you are having a chicken. So you are creating superbug, as a result of which you are becoming antibiotic resistant yourself. So people say that there is not far away the day when a common cold cannot be treated with antibiotics because we have developed so much resistance. How we can handle COVID in such situation? Crazy, right? So these are the different positions where antibiotic resistance, antibiotics work. They are called the DNA RNA synthesis. I talked about it. They talk about the cell wall, folate synthesis, cell membrane, protein synthesis, inactivating enzyme, target modification, immunity bypass, efflux pump. Everywhere I can generate an antibiotic resistance. Every step is important for the bacteria to survive. And keeping in mind a bacteria or a virus, including your COVID-19, loves to survive. They want to control the whole world. They want to survive by killing the host. They have no other job other than proliferate and survive. We have to be smart enough to outsmart them because their number is more. Our number is not so much. And we are getting killed by them. So they want to survive. They are mutating by natural pressure. That mutation we are not being able to stop. So you can only modify the drug, modify it, and try to see how you can survive further. That is the game we are playing all the time. And this is the inhibition of protein synthesis. This I talked about. This is basically inhibition of protein synthesis. This is a ribosome. Different drugs can act on 30S binding, 50S binding, tetracyclic pactomycin, hypomycin, etc. Tuberculosis I talked about. Robert Koch discovered and how this rifampicin completely not working very well. Still is not the best drug, but our burden is so high. We need to have a better drug for this. So if you look at it, these are 2006 slides, that first-line antibiotic, there are different kind of tuberculosis. We have multi-drug resistance tuberculosis, XDR tuberculosis. We have some different other kind of drug resistance. And these are the antibiotics people use for tuberculosis. And first-line antibiotics, these are the year they are employed. And they have more of them, all of them have antibiotics resistance now. It is true for everything. Even artemisinin, which is a infection, which is an inhibitor for malaria infection, it was such a wonderful drug, people thought. But now I hear in the Vietnam-Cambodia border, there are a antimicrobial resistance population. So it is coming up all the time, and I'm sure with the use of COVID, vaccine is important that way because you mutate and you inject. Influenza vaccine, or people tell me now, every 6 months you have to take a shot. In US, people do that for influenza vaccine because vaccine under the pressure of influenza virus, under the pressure of vaccine, they mutate. The binding site is mutant. You heard so much about spike protein interaction with HCE receptor, but those mutation sites are known. And if they mutate, then any vaccine which inhibit this interaction will not work further. So that's what. Remdesivir, I want to talk about it because this is a omics related issue. Meeting is a mechanism of action. This is an RNA-dependent RNA polymerase inhibitor. How remdesivir works? This is the structure of remdesivir. Sometime back I showed you the structure of nucleic acid bases. This is almost looks like ATP. Look at the ATP. You have seen the ATP structure I showed you a few slides before. In that structure, you will see this is the XONH2 group. This is a purine ring with a 5-membered ring attached to it. This is a ribose sugar attached to a phosphate. But see, this is a beauty. Remdesivir has an alteration at this position, has an alteration at this position, 5'. How the RNA synthesis goes on? RNA synthesis goes on 5' to 3' direction, where 3' OH gets deprotonated and attack the innermost phosphate of the incoming nucleotide, and the SN2 attack, and the phosphodiester bond is formed. That is a mechanism, trigonal bipyramid mechanism. I'll not go into the much chemistry on that. We have worked on this area. And it works. However, now you look at this. It was fortuitous, not designed that way. Many people have tried different modifications, nucleic acid bases. If I modify the base like this, or if I put aza here, aza nucleotide, then this compound cannot form a 5' 3' bond. However, this fits into very successfully, it fits into the pocket of RNA polymerase so nicely that it can, if I have this compound remdesivir and mixture of all NTPs, ATP, UTP, CTP, GTP for RNA synthesis in the reaction. So what is the reaction is happening? If I have a reaction mixture or a virus in the presence of a virus infection, HIV or OMIC, COVID, in the mixture I have DNA template or RNA template, is RNA-dependent RNA polymerase, so RNA template. It has all the 4 NTPs, magnesium, blah, 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 whatever is required for the synthesis. ATP, UTP, CTP, GTP are incorporated, RNA synthesis is going on. All of a sudden, I add remdesivir. Remdesivir will come. Its binding capacity and the concentration is more than ATP. So it will replace ATP and go and sit there. So it fits very well on the substrate binding site of RNA polymerase. And it binds so nicely and make phosphodiester bond that it can replace ATP. If it replaces ATP, then this molecule is not able to propagate further. Next nucleotide cannot come because it c't have a proper 5' 3' bond. So it becomes an inhibitor. So it becomes an RNA synthesis inhibitor. So this is basically the policy of how the drug manufacturing company are doing with partial success. There are other nucleotides that are coming there and playing a role there. So these are the inhibitors: rifampicin, soranidin, lipriamycin, tagifluoxacin, microcin J25, CBR702. And their sources are from different places. They are coming and they're blocking different steps of RNA synthesis or protein synthesis. For protein synthesis, mostly we are getting ribosome-mediated protein synthesis on RNA template and the synthesis of polypeptides. Now, this is the structure of rifampicin. One can ask me a question that how this big molecule become an inhibitor. This is a fortuitous observation by antibiotics because there are rich source of antibiotics coming from a lot of bacteria, and they have from soil bacterium and other cases. From there, the rifampicin was discovered. It binds to the active site of RNA polymerase so elegantly, like remdesivir binds to the RNA propagating elongating site. This goes and sits in the RNA polymerase active sites and inhibits one of the subunit of RNA polymerase, and transcription inhibition takes place. We worked on this for a very long time, and we showed how this whole thing worked basically. What is the binding site where we have found out that transcription inhibition happens. I'll not go and discuss about my work, but this is not the case time for that. But we worked in great detail how this inhibition can happen, how the enzyme structure is responsible for the binding of rifampicin and inhibition. And recently, this year, I think this year or last year, I forgot, I think this year, beginning of this year, a paper has come in JVC where they showed how remdesivir binds to RNA polymerase and inhibits transcription for the RNA by RNA dependent RNA polymerase. So these are all known and is structural coming, but these are not vaccines. These are natural synthetic analogs or chemicals which inhibit the process of enzyme. And guiding principle is this: inhibiting the enzymes pathways which are involved in this synthesis of macromolecules. And these are so important steps. Inhibiting them will become a very important parameters. So these are the kind of mixopyrin with RNA polymerase binding of rifampicin RNA. These structures are known by crystallography, cryo-electron microscopy, and areas are very well worked out. So this is basically the inhibitions. Now we know the detail further, how rifampicin can inhibit the synthesis by two modes of action in two different dyes. And these are part of drug discovery work today. And many of the young students, I can tell you with tremendous confidence, as the human population is increasing and different kinds of COVID teaches us various things. COVID tells us how to remain, how to swim in the ocean in spite of the pressure of death around us. And we are adapting continuously. And therefore, different ways things are coming up. And this is a very good area. As long as human being is there in the population, these are very important areas where science will progress. So there will be no dearth of work, no dearth of jobs in these areas. So students should think of newer pathways, newer ways, how inhibitors can be designed and can be worked out. Vaccine is, of course, one of them. So these are the structural parameters. I talked about it further. And now GNA has a unique ability, the guanosine, one of the bases, a unique ability to have a bending, to have a different conformations, G tetraplexes, different structures. And these kind of structures are so important for DNA. So it's a fascinating structure. But I showed you one of the slides before. The formation of these structures can be inhibited by different ways, which can be activated by these kind of chemical molecules. Now I come to something which I like very much. I still continue to work on. That upon infection, we inhibit the bacterial growth. We thought over, we are working on this for a very long time. We thought over a new area of working that in this work, what we wanted to discuss, I want to present to you, that we asked the question, my students and me, we thought about it about 10, 15 years back, that if we inhibit DNA synthesis, RNA synthesis, remember I'm talking about bacteria, not COVID, because virus cannot be handled this way. Whether they have the same pathway or not, we do not know. But what we thought, that there are small molecules which appear, which are guanosine derivative, PPG, PP, that means there is a guanosine moiety right here. There are two different phosphates, one is the 3', another is the 5'. Sometimes 5' have 3 phosphates, 2' have 2 phosphates. So one base has preponderance of phosphates group around it, 5' and 3'. These molecules, they are called stringent factors. If I grow a bacteria under starvation, no amino acid, no food, no sugar, nothing, bacteria still grows in a salt medium. But after some time, they show stringent response. At salt, you will see the bacteria is growing. And 50 years back, some scientists wanted to find out what is this. So they took the bacteria from those cells, crushed them, ran a paper chromatography, and identified two molecules coming up in a large in abundance. Analyzed the structure by chemical means and showed they are guanosine tetra, guanosine pentaphosphate. And they come as a magic. If you feed the bacteria, then bacteria with the sugar or amino acid at that time, they disappear. So they come and go like a magic, and they're known as magic spots, they are called at that time. Now everything has been opted out. We now exactly know how it works. It binds to RNA polymerase, regulate transcription. What happened? Bacteria, see how smart the bacteria is. When you have a problem of food, you have nowhere to go for food. Starvation signal. Human being will die, right? Most of us. What bacteria is doing is adapting itself, is adapting in such a way that under starvation condition, under stringent condition, they try to minimize their energy expenditure. That if you don't have food, what will you do? You sleep, called hibernation. Sit in a corner, don't do any activity. You keep your basal minimum multiplication process going on. Wait for the opportunity, wait for the good day when food will be given to you and you will survive further. That is called hibernation or stringent factor, stringent condition. And they survive. They found spores sometimes and survive years after years. So much so, it has been shown Egyptian mummy who died 3,000 years back, when you take out the lungs and culture, you find TB infection, which can be resurrected by adding proper medium. So therefore, a bacteria can survive like that, tuberculosis, even for thousands of years. So therefore, these are the very important points. So we thought, if these molecules are so important, can we asked the question, can we design an inhibitor like remdesivir, I showed you an example, or rifampicin which blocks RNA polymerase. Can we design an inhibitor which will mimic this guanosine and bind to a position where it will inhibit the formation further and inhibit the stringent response pathway? We worked, we had partial success. We know why the DNA replication, transcription, translation, how biofilm information, sporulation. Every one of them we have looked at and looked at how they are working, how they can be inhibited further. And we went ahead and further with this. And we showed with a series of publications over the last 10 years that both the synthesis of this molecule, pentaphosphate, cartoner, and the degradation in mycobacteria, done by a protein which has several organisms or several domains, and these domains are very important for the transcription, for the stringent response inhibition. Similarly, recently we found that there is a homeostasis. There is an N-terminal domain and C-terminal domain of the protein. One synthesizes the protein, another degrades the protein. They work together to generate guanosine tetraphosphate. We ask the question, can we impair with this? Can we play some role to inhibit these particular steps and proceed further? So do that stringent response modulation we can do or not. Again, we worked on this. Recently, we published this year a review on this, that we have a hydrolysis synthesis and different domains. And we have taken these different domains and tried to inhibit one of these domains by an inhibitor and see their functional implications. Similarly, we have worked on the different mode of these enzymes, how the different parts coming together, etc. And we know the whole mechanism of action. And if we know the mechanism of action, the role of individual subunit, role of individual substrate, GMP, GTP, GTP, we can look at the whole process and we can see how the inhibition can work. So therefore, our main goal was, and even now today, can we design an inhibitor for this stringent response pathway? So second messengers in bacteria, these are called second messengers. Why? What is the meaning? I'll discuss the term. If you have a food shortage, temperature, pressure, environmental condition changes, these are called first messenger, which gives a message for bacteria that you should take precaution It's probably acting in virus too. You take precaution. They relay that message inside the cells to their neighbors called cyclic di-GMP, and these are second messages. And these are the second messages: cyclic di-MP, cyclic di-MP, PPBGP. These are second messages. They work together to maintain a particular state. And they have a particular pathway of synthesis and degradation. We worked for a very long time on these matters. I will not go very much in details. However, again, to make students keep interested in this whole area, I tell you that few years back, scientists find out that bacteria showed another property called quorum sensing. What do you mean by quorum sensing? Quorum sensing is the one which says that a bacteria understands that there is a danger. It passes through chemical means, these kind of second messages, relays the message to the neighbor telling that be careful, something has happened. So they act in concert, they work together. So their whole process of activation, whole survival mechanism is working together. It's called quorum sensing. And this is a very important phenomena. So if we impair with the quorum sensing phenomena, we can make bacteria to die individually. Bacteria is a social animal. I'll talk about it tomorrow in great detail. That's why I want to talk about social behavior. You will see that bacteria, minute and minute organism, perhaps virus also, you do not know yet, that they communicate. The language is through second mass chemical language. They have altruistic behavior. In a colony, if there are too many of them and death is inevitable, the poorer ones die first to keep the better ones are alive. They also cannibal, they eat each other in under tremendous pressure. So every single social animal, whatever behavior they show, bacteria show the same behavior, and my tomorrow's talk is on the basis of that. So I'll not talk further about it. So domain architecture of these proteins which involve in second messenger synthesis and degradation goes on like that. And we showed recently that how these two domains of a protein which synthesizes cyclic di-GMP play around. These are the two domains, N-terminal and C-terminal domain. They interact with each other to play around to make a second messenger keep on working and running. So we have a heterogeneous population of cyclic di-GMP and GTP. So these are all coming from GTP pool. They make guanosine tetraphosphate or cyclic di-GMP. And heterogeneity in the distribution in bacteria happens on the basis of that. So these pathways are very good pathways to control their propagation, control that not much work has been done. No commercial medicine is available on stringent response. But when I finish the talk very soon, I'll show you some very interesting observation I found recently, and I'll keep you posted on that as I proceed further. So our main aim is to get an inhibitor which will interfere with the bacterial stringent response in different direction. So you ask the question, does PPBGP or cyclic di-GMP regulate antibiotic sensitivity? If I remove this, the antibiotic sensitivity will go. It's an obvious question to ask. This is OMIC study, and we call it phenotypic microarray OMIC study. So what we did, we use a machine which I have in my lab. It's called phenotypic microarray OmniLog machine, where I can look at the growth and disappearance, growth and death of bacterial colonies in the presence of different concentration of antibiotics in different growth medium, where I don't need to shake the bacteria for growth. It is a static growth. Oxidation happens of particular dye, changes the color, and the color is sensed by this as a function of growth. So you look at the growth of organism by looking at a coloration, changing color profile, change in OD. So there is a metabolism with time, there will be one color. If there is a reference, it will go like this, and in the inhibition, there will be another color. So a difference in color, we can look at the growth and the death of organism. So these are the kind of plots we get, and we can together 96-well growth plate. So I can do tremendous amount of bacteria under different condition, growth and death in the presence of antibiotics. We did this work at long period of time, and we have some analysis of the result. I will not go very much into details. But if we looked at the antibiotic knockout, antibiotic sorry, the genes responsible for antibiotic metabolism knocking out the PPUGP synthesizing gene and cyclic diGMP gene and second messenger genes and look at the antibiotic sensitivity so that we can correlate directly with the antibiotic sensitivity and the presence of the second messengers. This is basically the validation of the data happens this way, and we get a several growth curve under different conditions. Presence of ampicillin, rifampicin, tetracycline, norfloxacin, streptomycin, erythromycin. Growth curve with a typical RT-PCR analysis, and we look at the growth of the organism, growth of the bacteria with the presence of different antibiotic conditions. What goes on like this, and we have almost solved why there are different resistance coming, reasons for antibiotic resistance coming. And we look at the proper growth phase the following way. So I'm coming close to the end of my talk. It is already more than 1 hour I am speaking. So the different kind of bacterial colony culture will be here, I have shown here. And they show the shape and size, how they changes under different conditions. So how these, these are new angle are coming. I told you the packing of the chromosome, packing of the DNA. We have seen the cell morphology changes, cellular morphology changes in the presence of antibiotics. Biofilm formation, overexpression of PPUGP gene or overexpression of cyclic diGMP, how they changes. So all this creates a tremendous areas of research for us to look at how the organism changes in the presence of antibiotics. So they change the cell length also. I think I don't want to continue further on this. And we have looked at, and so therefore the second messengers have a havoc in the cellular morphology, multiplication, dimerization, length, and then metabolism at the basic enzymatic level. Cell wall has several such different ways to look at inhibition. Glycolic acid, arabina, mannose formation. It's a very complex cell wall structure. I have not discussed that because it's a very complex science by itself. So for developing an inhibitor now, what we did, this is the last part of my talk, what we did here. I told you, remember, that analog of a nucleotide, remdesivir is a classical example of that. I include these slides because I talked about remdesivir. Stringent response inhibitor cannot generate a PPUGP analog. Lysine is a well-known inhibitor, but it is very high concentration. So what we did, we developed a modification of acylated modification of the nucleic acid base, PPUGP, guanosine moiety, and showed it that it has a tremendous inhibitory ability. We have published this work in 2017. It has a lot of demand in the literature, and benzoylated acylated compound. At that time, I was not even able to do tuberculosis work in my laboratory. So I collaborated with Christina Stalling of Washington University, USA, and we together published this paper in Antimicrobial Agent and Chemotherapy, how the biofilm inhibition happens with benzoylated and guanylylated nucleotide tetraphosphate. Second point I would like to see, when I was doing this work, a very small observation came, which was amazing. Again, remdesivir, when I told you about structural analog. If you look at vitamin C, ascorbic acid, its structure has tremendous familiarity with the sugar moiety of the guanosine tetraphosphate. A chemist in the group who are audience will understand immediately what I mean. This has a double bond, but this has no double bond. So there is a geometry of the carbon here, planar here is not. But this has tremendous structural homology of this. So we thought, me and my student, can we look at vitamin C as an inhibitor for stringent response? Lo and behold, it does. And vitamin C in large, and see, vitamin C is such a drug. It's such a vitamin, it has no adverse effects so far, a very high concentration. So you drink lemon juice, orange juice as much as you can. Can you take care of it? You develop your immunity. Can you take care of the stringent response? Yes, we do. We saw the assay and it worked very well. And as it inhibits stringent response, it inhibits the growth of microbacteria. I strongly believe vitamin C will have a tremendous effect on the. I do not know the chemistry, so don't quote me for this. But vitamin C, vitamin D will have a tremendous good effect, even for the COVID infection. Even it doesn't have, by boosting the immunity is enough for it. So if you go on drinking orange juice, it will be wonderful for the health in future, for any bacterial or viral infection. So these are the PPVGP, this is the molecule. We have two inhibitors designed by looking at the structural analog, acetylated benzoylated derivative, ascorbic acid. Both are published. I not patent them, but I am still supplying the materials and people are interested to take it further. So I'm not doing this work for you. So I stop my presentation here. I hope I kept you interested in the process. But I'll talk about these processes further again tomorrow in the midst of social behavior of bacteria. Thank you very much.
Thank you very much, sir, for your insight on the subject of antibiotic resistance. I hope that as future scientists, we dwell on this topic more actively and hopefully find a solution soon. We have a few questions. We'll take them now. The first question is from Arun Kumar E. If probiotic strains used by commercial manufacturers tend to carry antibiotic resistant genes, can this lead to antibiotic resistant gene transfer to our gut microbiota? What will this affect?
Yeah, there is a possibility, but there are ways to control it also. Many times probiotics are not allowed to have those genes and they're modified.
Okay, sir, thank you. Moving on to the next question by Soumya Nenan. Do you think there will ever come a point where the use of antibiotics becomes obsolete due to the increasing antibiotic resistance in microbes? What could be the alternatives to combat this?
Very good question. Superb question. I am certain your question will have a very negative impact. It is indeed going to happen because we will develop in some time if we don't use our restriction. Like climate change, if we don't push restriction of antibiotic usage internationally among citizens and limit it unless dire condition happens, this is going to happen. And then people will stop using antibiotics, there is not work. What is the consequence? Then that's why I talked about any other inhibitory pathway. We cannot develop a vaccine so easily. So any other way we can do that. So newer method, newer science will come, I'm certain. But the fight is on all the time. Bacteria tries to survive, we want to survive too.
Thank you, sir. The next question is by Sneha. Do you think if antibiotics are sold only if there is a prescription by a doctor, is there a chance of the resistance acquiring rate going down?
Very much so. Very much so. It should happen that way. Going down, we are not eliminating it, but going down because we are not indiscriminately using antibiotics. If you have a common cold, which will go by just resting, you don't need antibiotic. Many times viral infection, people give antibiotic to resist the secondary infection, which is important, but not always.
Thank you, sir. The next question by Arun Kumar. Do you think
Yes. I'm losing the audio. I'm losing the audio.
Can you hear me, sir?
Now I can hear you.
Hello?
Yes, I can hear you now, but it is cracking.
Okay, so I'll repeat the question. Tell me if there's any problem. Do you think employing bacteriophages for superbugs might be a possible solution in the future?
Yes, very much so. Thank you. Very good question. This was used long back for tuberculosis. People used to use, or for Salmonella typhimurium, a typhoid infection, there was a P22 bug. People used to give phages. Bacteriophage inhibit the synthesis of inhibition of the bacterial culture. In Bangalore, there is a company called Gangajan is doing that. And there are many attempts, but for this is called phage therapy. But one has to be very sure about the science behind it and what pathway you are impairing. If your pathway is very well predicted, is well formulated, then of course this is a very good science. One can think of it. Bacteriophage therapy.
The next question is kind of like a follow-up by Larissa Gomes. Rather than bacteriophage itself, what about using gene responsible for lysis of the bacteria, that is endolysin? What are your thoughts on that?
Very good pathway again, and people are attempting that. It really works, but it should be specific for the bacteria, right? It should not create problem for the host. See, the advantage is, let me point out it again. A tuberculosis, suppose, infects human being. Rifampicin is a classical inhibitor which inhibits tuberculosis RNA polymerase, specifically binding it. We know where it binds, how it does, and it kills tuberculosis. But rifampicin will not touch human RNA polymerase. Eukaryotic RNA polymerase, it will not do anything. So it's a classical drug. It's a textbook example. But host is not affected, but the bacteria is affected. Not always so true for many cases.
Moving on to the next question by Dr. Poppy Datta. Does antibiotic resistance go away with time?
It will go away with time is another very good question, only comes from a doctor. If you prescribe it and you make strict vigil, UN sponsor, WHO sponsor, strict vigil, there is a punishable offense if you buy antibiotic without prescription and you throw away half it. You buy only the complete dose. Then with such strict regimentation, within 5 to 10 years time, it will go away. It will reduce very much. Resistance will go. Bacterial population will not be able to develop very well. But it requires tremendous monitoring. And that is very important. But I hope that only that can come by the strict measure. In fact, in some countries, people are thinking for poor people. If you take your complete dose, dose will be given by a doctor in a hospital, and along with the dose you give medicine with some money. And this was initiated in US by President Clinton time for tuberculosis patient, that you not only get an antibiotic, you also get some money. For the money, you will come back for the second dose, even if you are okay. So these are social implications, very much implications. It's called dot. Yeah, go ahead.
Yes, sir. Sorry. The next question is by Dr. Beatrice Sequeira. Antibiotics may not reach very deep wounds in the case of Pseudomonas aeruginosa infections with biofilm formation. Right. So we might not be able to increase the concentration as it may cause toxicity. So what kind of method should we adopt?
Yeah, this is a very good question, also very very knowledgeable question. The MRSA, like methicillin-resistant Staph aureus infection, biofilm formation, catheter biofilm, heart transplant biofilms, is a notorious thing. And we have worked on biofilms. I have worked a lot on biofilms. These biofilms are matrix through which nothing can propagate. So what we thought, we are working still on it, that maturation of the biofilm, at the stage of maturation, formation of biofilm, you incorporate such inhibitors which will not allow it to form biofilm or make a fragile biofilm. Fragile biofilm means it will crack with a little provocation. And these are the ways. The research, these are very active research field, how biofilm formation can be impaired. But you are absolutely right. Biofilms is a nuisance. Antibiotics cannot pass through.
Thank you, sir. The next question is by Ms. Shalmali Kamath. Does development of antimicrobial resistance depend on the type of antimicrobial mechanism? That is, are some antibiotics more likely to become ineffective because microbes develop resistance towards them faster?
Absolutely. That's the whole point of the talk I told you, that if you all the time look at central dogma of molecular biology, DNA and protein, that's so important. Bacteria will adopt a first-time mechanism of mutation there. We do not know how. That's why I come to stringent response.
Thank you, sir. Our next question: can natural vitamin C be replaced with chemically synthesized if the dosage consumption to fruits is not enough?
Yeah, but people did not try very much. It's possible because so much vitamin C comes in abundance from cheap fruits. See, not everybody has to drink orange juice, musumbi, lemon. They have so much of vitamin C that you get a good amount of concentration from eating them. But it is very good. Linus Pauling, remember the best chemist of the last century. He said vitamin C is the only treatment for common cold. So it has so much effect. And now we are showing it in the case of bacterial infection. I do not know about viral infection, but it's playing in my mind. I do not work on COVID. But I suggested to many people that doctors should work on vitamin C who are handling COVID, how it affects viral infection.
Thank you, sir. Our next question is by Snesha Baumkar. If coronavirus succeeds in killing the human race, will it disappear too, or will it mutate itself to infect any other living organism?
It will not be able to, don't be so negative, it will not eliminate human race. Before that, they will die. But what will happen, that we are learning to live with it. We are new in it, but I'm certain we'll win over it. So I'm not even thinking about their killing human race. They will not be able to possible. They cannot kill human race, but they will make a substantial mark like they did 100 years back.
Thank you so much, sir, for so patiently answering all the questions. And
I hope
with
people
this,
have enjoyed it and they are attentive.
yes, I think I speak for everyone. I think it was a very, very good talk, and we've gotten a much better understanding into this topic. With this, we've come to the end of the second session, and I thank all the participants for their questions and actively being involved in the session. Over to you, Kaljada.
Thank you, Madiha. We will now break for lunch. Participants are requested to stay logged in, and we'll be back in the meeting after a break at 1:15 p.m. Thank you all.